So this talk is really geared towards people who are kind of new to cystinosis, so especially any newer families that are here, just to kind of give you the basics and framework for the rest of the stuff you'll be hearing about at this conference. So I apologize to the many of you who have heard this talk or a version of it before. A lot of the slides are the same, so bear with me on that. Like Denise said, I have two kids with cystinosis. So this is Sam when he was 1 year old. This was on his first birthday. And right around that time is when he was diagnosed with cystinosis. So like all of you, had a similar story, wasn't growing, wasn't eating, wasn't making milestones, was sick all the time. Took us several months to get the answer. And then a couple months after that, Sam had to get his feeding tube, a gastrostomy tube, because he just still couldn't maintain nutrition and hydration and giving all these medications around the clock. So this is a picture of him in the hospital after his surgery, and he was miserable, but he's still trying to smile, being a good sport about things. And Sam really didn't eat anything for like 3 years. He drank a lot of water, but he just wouldn't eat. You tried to give him food and he would just immediately throw up because he had such a strong gag reflex to all food. So I thought, man, Sam he's never gonna eat. He's just gonna live with this pump for the rest of his life. And he was on that thing 24 hours a day. So it was tough. But around the time that Sam turned 2 years old, things kind of started to stabilize. We got used to this new life of giving medicines around the clock, being hooked up to this feeding tube all the time. And we thought, well, we'd really like Sam to have a sibling. So, 75% chance that I won't have cystinosis. Let's see how it goes. And so we were fortunate to get pregnant and have another baby 9 months later. When Sam was 3 years old, when Lars was born. And we got Lars tested right away, and he also has cystinosis. And it was a blessing that we were able to get that early diagnosis with Lars and get him on medications right away, the Cystagon. And so he's done really well. He doesn't have as much kidney damage. And so this is another picture of little Lars. He's got that nice blonde cystinosis hair, And this is Sam and Lars now. So they're 9 and 12. Lars is the goon on the right, and he's just as tall as Sam now. They're best buddies. Sam may not admit to that, but they do everything together, and it's been really fun. And Sam does eat food. He eats a lot of food, and he was actually able to get his G-tube out a month ago, or back in January. So that was a really, big moment for us. We're very proud of him. He takes all of his medicines by mouth and did that for like a whole year just to prove he didn't need that G-tube anymore, and now it's gone. So it's just really cool that as we keep moving, things get better. So to understand cystinosis, you do need to know a little bit of cellular biology. So this is a picture of an animal cell that I stole from the internet. But basically you can see there's all these compartments within the cell. There's that big blue sphere in the middle, that's the nucleus. Then there's these other blobs that do other things. And each of these compartments has a special job in the cell. There's a lysosome, there's the Golgi apparatus, there's ribosomes. But really probably the most important organelle of the cell is the nucleus, because that's where the DNA is. And DNA is that genetic blueprint that every cell in the body has the same genetic blueprint. So that DNA is this green stranded thing that's wound up it's called a double helix, and it's wound up very tightly into these structures called chromosomes. So the big blue X thing is called a chromosome. And you have 23 pairs of chromosomes. Half of them are from your mom and half of them are from your dad. And this is what they look like under a microscope. But what is DNA exactly? What is it really there for? So like we say, it's this genetic blueprint, but it's really like a library of instructions for how to build different proteins. And so the cell has special enzymes that can go in, and if it wants to build a certain protein, it'll go into this library and unwind part of that chromosome and find those instructions, and it'll copy them down into a little transcript called RNA, and then that transcript gets fed through some special machinery in the ribosome that will create a protein. Okay, and a protein is made up of building blocks called amino acids. So the way that we explain this to my kids is with Legos, because they are obsessed with Legos. And it's a nice analogy, but basically, the instructions for building your Lego set, that nice booklet it comes with, that's DNA. And then you got a master builder here. Maybe many of you have seen the Lego movie. This is Emmett, the master builder. He's the ribosome. And then all the little pieces that come in the bag that you need to assemble, those are your amino acids. And so now you have instructions for how to build this nice protein. In this case, a fancy race car with a spoiler on the back. So what happens then if part of those instructions are missing? We call that a mutation. So now, when the ribosome is reading these instructions and trying to build this race car, it doesn't have all the instructions. So it builds a race car that looks pretty cool still, but you'll notice now it's missing that spoiler. It doesn't have the instructions to do that. And so that's what's going on in genetic diseases, where there's a mutation in the DNA that's causing the cells to build a faulty protein, or sometimes they don't build the protein at all. Okay, so we talked about how every person has 23 chromosomes, or 23 pairs of chromosomes. Back in 1998, Dr. Corinne Antignac in France discovered the location of the cystinosis mutation. And it's on the 17th chromosome. And one of the scientists that worked in Dr. Antignac's lab during this time and on this project was Dr. Stephanie Cherqui. So this is a picture of the 17th chromosome. And there's a little arrow there that shows you where that mutation is found. And since that time, hundreds of mutations have been found in this gene. But the most common mutation is called the 57KB mutation. There's 57,000 base pairs of DNA that are missing. And so that's the most common one you'll see in cystinosis. About up to 75% of people of Northern European ancestry have this mutation. But there's lots of other mutations too. So cystinosis is an autosomal recessive disease, which means that an individual needs 2 copies of a mutated gene in order to have the disease. So if you only have 1 copy of a mutated gene, then you are a carrier. So for the parents in the room, that's you, right? And so every time you have children, there's a 1 in 4 chance that that child can have cystinosis. But there's also, 1 in 4 chance that child won't have any mutated genes at all. They'll just have 2 normal copies. Then there's a chance that they could get a normal one from mom and a mutated one from dad, and that child will be a carrier like you. They could get a mutated copy from mom and a normal copy from dad, and that child will also be a carrier. Or they could get a mutated copy from mom and a mutated copy from dad, and then that child will have cystinosis. So you'll notice that I use 2 different shades of blue here, and maybe you can tell that on the screen, but you don't have to have the same mutation. So my wife and I, our kids have had their DNA sequenced. We have different mutations, but a lot of people may have the same mutation, but just wanted to point out that it doesn't necessarily have to be the same one, because there's hundreds of different versions of that mutation. So this is a picture of kind of a schema of what the cystinosin protein looks like. This is the protein that is built from these instructions in the gene, and all those little balls are amino acids. So this protein lives in the membrane of something called a lysosome. And so you'll remember that there's all these different compartments in the cell, and one of them is called the lysosome. And you'll see the lysosome is those little circles with red dots in them in the cell. And really what the lysosomes are, they're like the recycling center for the cell. And they're kind of like a big bag of digestive enzymes. And what happens is stuff gets engulfed by the cell, like food particles or like foreign invaders, or just like old proteins that need to be recycled. Those things get packaged up in a different blob, which then merges with the lysosome, you can see in this picture. And the lysosome spills all of those digestive enzymes and basically chews up those proteins, food particles, bacteria, whatever is coming into the cell. So, like I said, one of the things the lysosome does is it recycles old proteins. So in this slide, we have this orange circle representing the lysosome. And it can take up these old proteins like this old Lego race car. And then you have these special digestive enzymes here represented by Lloyd, the green ninja. And he comes in and he chops up that race car into its different parts, all these amino acids. And so each of these amino acids then can leave the lysosome through a different transporter. See, the red one goes out there, this blue one goes out through that blue transporter, etc. And I think I made way too many animations here. Okay, but you'll see that each amino acid has its own transporter protein, and that's very important. So in the case of cystinosis, there is an amino acid called cysteine, represented by this blue Lego, and it gets out of the lysosome through a transporter called cystinosin. But in the case of cystinosis, there's a mutation in the instructions, so it builds a faulty protein that either isn't there or doesn't work. So now that cysteine tries to get out of the lysosome, but it can't. And so then it just accumulates within that lysosome until there's too much, and it can cause crystals, and it can damage the cell. And this is going on in every single cell in the body, all 40 trillion of them. And that's why cystinosis affects every organ and every tissue in the body. So fortunately, back in the 70s, a very smart doctor named Jerry Schneider at UCSD discovered a treatment or a drug called cysteamine that actually reacts with cysteine. So it goes into the lysosome, and it binds to the cysteine, and it forms a new molecule that looks a lot like a different amino acid called lysine. And that now can get out of the lysosome through a different transporter protein. So that's how cysteamine works. It goes into the lysosome, binds that, helps it get out of the lysosome. So this was obviously a big groundbreaking treatment for people with cystinosis. Back in the 80s, they did the drug trials back at NIH with Dr. Gall. And then in 1994, I believe, is when it was FDA approved. And the first version of it was Cystagon, which I'm sure many people in the room are taking Cystagon. You take it every 6 hours, which means lots of sleep interruptions. So my son Samuel was on that drug first until he was about 2. My son Lars was on it until he was about 5. And everyone, I think, is familiar with the side effects of Cystagon. It causes a lot of gastrointestinal symptoms, nausea, vomiting, abdominal pain, and the sulfurous odor. So then in 2013, there was a new version approved called Prosisby, which is a 12-hour formulation of cysteamine, which was developed with CRF-funded research. And so this is basically taking that cysteamine and coating it with beads so that it doesn't dissolve right away. It gets past the stomach. So the beads are designed to dissolve in a basic environment in the small intestine. So that delays the absorption of the drug and delays a lot of the side effects too. And importantly, because it's designed to dissolve in a basic environment, it should be taken with something that's hopefully acidic. So when my son Samuel, who I should point out, he's up here on the front row because he's an A student. This is the first time he's come to see me give a talk, so it's kind of cool. But when he was 2, we got him enrolled in the RP103, is what it used to be called, the Prosisby trial for phase 4. And he was one of the first, he was the youngest kid at the time and the first to do Prosisby through a G-tube with his friend Henry Sturgis. So we had lots of fun that first morning trying to figure out how to get all those beads through the G-tube, immediately clogged it. But we worked through it and figured it out. But Sam was able to get, I think that was in 2012, and then we were very lucky to stay on that medication when it was FDA approved. I think all of you know that it's an expensive medication. When I looked at our recent insurance claims, It looked like it was about $112 a pill. So I'm always delighted when I find one at the bottom of my car somewhere where it's just fallen under the seat. Like, hey, look, $112 pill, Sam. No big deal. But very expensive medication, and so we're grateful to have it. So when the pandemic hit, we had to go to remote learning for a while, and so we thought it would be fun to do some science experiments with the kids. And so we had Sam and Lars ask this question: what would be the best drink to take with your Prosisby in the morning? And we know that you want to take it with something acidic, so we had them choose different drinks at the grocery store, and we had them kind of predict what they thought would be the best one based on pH. We bought some little pH strips so we could test them, and here are some pictures of them doing this experiment. So what do you guys think is the most acidic drink on this chart? I hear some orange juice, Coca-Cola. That's pretty acidic. Okay. What do you think is the most basic thing on this list? The water? Do you think bottled water or tap water is more basic? Bottled water? Do you think that, okay, and then do you think water or milk is more basic? Water? Milk? Who says milk? I always thought milk was really basic. Who says water? Okay, so this was like very illuminating to me doing this experiment with them. Whoop. So tap water was the most basic thing we tested, and I'm sure this is based on where you live, right? Some people are going to have different levels of alkalinity in their water, but our tap water was extremely basic, and that's what Sam takes his Prosisby with most of the time. So that was illuminating. Bottled water was the most neutral, and then milk, I was surprised, is actually fairly acidic, and so, and then you'll see that Coca-Cola, orange juice, apple juice are all pretty acidic. In our test, orange juice was more acidic than Coca-Cola, but I think when your pH is that low, it probably doesn't make a big difference. So anyway, just something to be aware of when you're selecting what beverage you're taking your Prosisby with. I wouldn't take it with milk because the protein and fat in milk will impair the absorption, but taking it with tap water is probably not the best idea. Although, if you're taking it with tap water and your levels are okay, then that's all right. Ultimately, you just want to do it the same way every time, especially on the day that you get your levels tested. Make sure you're taking it with the thing you would normally take it with. So like we talked about, cystinosis affects every cell in the body. So because of that, it's going to affect every organ and tissue as well. And really the first thing that we notice is its effects on the kidneys. It's going to cause something called Fanconi syndrome, which Dr. Grimm will talk more about. But this leads to that excessive urination, excessive thirst, and loss of electrolytes in the urine, which causes low electrolyte levels in the blood. And then over time, this will progress to kidney failure. We also see that it affects the eyes, so we see deposition of crystals in the corneas. This is happening pretty early, and so children can actually be diagnosed just with a slit lamp exam. Even at the age of 1, they'll have crystals in their corneas, and as we know, this is treated with the drops. Another common early manifestation of cystinosis is rickets because of loss of phosphorus in the urine, and so they get hypophosphatemic rickets. They also have short stature just because of nutritional deficiencies. And a common bony malformation is this valgus deformity, which is like a knock-kneed. But we are learning more and more about the way cystinosis affects the bones through research funded by the CRF. We've learned that it's not just about electrolytes and minerals and vitamins. There's also a lot going on at the cellular level with the way the bones are built and broken down. Another common thing early on is gastroparesis or dysmotility. That's something that's very commonly seen in children and adults. Around the age of 5, children may develop hypothyroidism because of deposition in the thyroid, so that's something to be aware of and to be testing. There are lots of neurological complications of cystinosis, but one of the more common ones is elevated pressures in the brain. Intracranial hypertension can cause something called pseudotumor cerebri. So if your child's having headaches, that's something to be aware of to get checked. And then Both men and women will have hypogonadism and delayed puberty. Many women have been able to have children. Men are technically sterile, but there are procedures that can be done to retrieve sperm from the testes directly and then used in vitro to have children. So that is a possibility. But because of that delayed puberty too, it's good to be aware that they may need to see an endocrinologist, and some children require some hormone replacement. And then a later complication of cystinosis is in the muscles themselves, so you can get a distal myopathy, which is muscle wasting in the hands and other distal muscles, and then that also affects the muscles of the pharynx, so it makes it difficult to swallow. And this is something that we are doing a lot of studying on. Back in Boston, Dr. Reza is doing a CRF-funded study on this. And then really it affects all the organs eventually, so heart, liver, pancreas, lungs can all have different manifestations of cystinosis. So because cystinosis affects every cell, you really do need a therapy that targets every cell. And so that's where Dr. Cherki comes in. This is a picture of Dr. Cherki with our boys back in 2019 at the last conference. We were able to go down and visit her at her lab and see where all the magic happens. And Dr. Cherki has been doing research on cystinosis for like over 20 years now. And one of the first things that she did after they found where the gene was they needed to create a mouse model of cystinosis. And so using special technology, they silenced the cystinosin gene in a mouse, and that's called a knockout mouse. So now this mouse has cystinosis, and then you can study and do research on that mouse to see if different therapies work. So the first thing that Dr. Cherki tried was allogeneic stem cell transplant. And this is where you take bone marrow from a healthy donor and give it to someone with the disease. And so they took a mouse that had the same genetic background as the knockout mouse, but had a normal functioning cystinosin protein. They took its hematopoietic stem cells and then gave it to one of these knockout mice. And that cured the knockout mouse, so they no longer had the features of cystinosis. And so this was a huge breakthrough when Dr. Cherki published that, and really led to the current stem cell trial. But when you do an allogeneic stem cell transplant, it is more dangerous because you're giving someone else's bone marrow to the patient. And so there's a risk of rejection or graft versus host disease, and there's lots of different complications that can happen. And this is a very effective treatment for a lot of other conditions, including a lot of blood cancers. But in the case of cystinosis, Dr. Cherki wanted to find a safer way to do this. And so that's where she came up with this autologous stem cell transplant, where you actually take the bone marrow cells from the patient or from the knockout mouse, and then you insert a corrected copy of the gene using a virus. So now these stem cells have the corrected cystinosin gene, and then you give it back to the same person. It's the same immune system, so it won't reject, and then those bone marrow cells can engraft very easily. And when she did this in mice, it had the same effect as doing the allogeneic transplant. So that is the study, the experiment that has led us to the human stem cell transplant. Here's a picture of Jordan, who was the first person back in 2019 to get that. And really, this is the treatment that we've been, that we hope will work because it can target every single cell in the body. Like we talked about, this cystinosin gene is not working in every single cell, so you really need a way to deliver that to every cell, and we think that this is the way to do it. So that's all I have. This is a picture from, I think, 2019. It's nice to see that the kids are still crazy this year. There's not as many of them this year, but hopefully we'll be able to have everybody come back next year. And that's all I got. Thanks.